I am Chengxin, an HHMI postdoc from Yale and a participant of CAFA. CAFA is one of the most important community-wide challenges for protein function prediction. Evaluating function prediction is complicated due to incomplete ground truth, imbalanced classes, and structured outputs. This work discusses our proposals to address some of these issues. In CAFA, the organizer preselects around 100,000 Swiss proteins from the reference proteome of around 20 species. The participants are given 4 to 5 months to generate results for all protein targets and submit the result by time t0. After a growth period of 9 months at time t1, a subset of a few thousand proteins get new experimental annotations. They will be used as assessment targets. We can define 3 kinds of targets. A no-knowledge or NK target does not have all annotations at T0. A limited knowledge or LK target has all annotation at one goal aspect T0 and get new annotation at another goal aspect at T1. A prior knowledge or PK target gets new annotations at the same goal aspect where it has all annotations at T0. CAFA only considers no-knowledge and limited knowledge targets, and mainly focuses on no-knowledge targets. Prior knowledge targets are ignored even though there are more than no-knowledge target and limited knowledge targets combined, as shown in this table. Additionally, CAFA dataset only includes 4% of species and 25% of no-knowledge targets from the full UniProt. For more comprehensive assessment, we propose the evaluation of all 3 target types. To include as many targets as possible, participants with live-weighting methods and sufficient resources can submit predictions for the full Swiss prot, reference proteome, or even the whole UniProt. Alternatively, participants can submit standalone prediction methods, ideally in a containerized format, such as a Docker image, which is the main submission format for the DREAM challenge. Submission of methods ensures reproducibility and avoid wasting resources for predicting pre-select targets that are not eventually evaluated. Another issue is baseline methods implemented by CAFA. A baseline method uses simple statistics and heuristics. Simple baselines are not necessarily bad. For example, as shown in this plot for CAFA2 CC prediction, the naive baseline in red outperforms many top methods, even though naive predicts goal terms only by their background probability in the training database, regardless of the target protein. The more problematic baseline is BLAST, which predicts goal terms by the maximum local sequence identity among templates from a default BLAST1. It is problematic because the score can be inflated by an unrelated template aligned to the target by a tiny portion of the sequence. As shown on the top for Fmax and at the bottom for weighting Fmax, BLAST local sequence identity is even worse than copying all computational annotations, such as IEA terms, from the UniProt GOA database. We propose another BLAST baseline called BLAST frequency, which counts the number of BLAST hits with a goal term among all hits. Without changing any certain parameters of BLAST, This frequency-based score consistently improved the blast baseline. Our poster additionally shows that blast frequency also outperforms E-value, bit score, global sequence identity, and ranking. Another observation from this plot is that despite prior existing annotations, prior knowledge targets in white is actually more difficult to predict than no knowledge targets in black and limited knowledge targets in gray, especially for biological process goal terms in the two central plots. This shows that prediction of prior knowledge target is not trivial and should be considered in CAFA. With that, we will thank the participants of a CAFA town hall held earlier this year, where we get many of the ideas for this project.